Number 1. Helicobacter pylori, the 100,000-year-old stomach colonist. The human stomach is one of the most hostile environments in the natural world. Hydrochloric acid drops the local pH below 2. Pepsin enzymes break down proteins. A thick mucus layer shields the gastric wall. Immune cells patrol every fold of tissue. And yet a single spiral bacterium learned how to live there, set up permanent residence, and ride along with our species for tens of thousands of years. That bacterium is Helicobacter pylori, and it does not float freely in the acid. It colonizes the mucus layer that sits directly over the gastric epithelium, drilling into a thin protected zone that most other microbes cannot reach. Genetic evidence places this organism deep in human prehistory. Strain phylogeography, the practice of mapping bacterial lineages onto geography, suggests that H. pylori was already associated with humans before the major out-of-Africa dispersals. Researchers including Mark Ochtman and Bodo Lintz have published work showing that distinct bacterial lineages mirror human continental ancestry. The labels are not poetic, they are descriptive. There's HP Africa 1, HP Africa 2, HP Europe, HP East Asia, HP Asia 2, and several subgroups within each. Each lineage tracks a population movement. When humans walked, the bacteria walked with them. Transmission happens early in life. The bacterium typically passes between family members during childhood, especially in households with limited sanitation, shared utensils, or crowded living conditions. Once acquired, it can persist for decades. To survive the acid, H. pylori produces an enzyme called urease, which converts urea into ammonia. The ammonia neutralizes acid in a small cloud immediately around the bacterial cell. The spiral shape and whip-like flagella allow it to drill through mucus and reach the protected layer above the stomach lining. Most people who carry the organism feel nothing. The chronic active gastritis it produces sits below the threshold of awareness. But persistent inflammation disrupts mucosal defense, alters acid regulation, and over years can lead to peptic ulcers. Two virulence factors stand out. KGA, the cytotoxin-associated gene A protein, interferes with the host cell signaling pathways. VAC-A, the vacuolating cytotoxin, injures cells, modulates the immune response, and damages tissue. Strains carrying these factors are linked to the most serious outcomes, including non-cardia gastric adenocarcinoma and gastric malt lymphoma. Adult global prevalence still hovers around 40 to 50%, with sharp regional inequality. The modern failure point is antibiotic resistance, particularly to clarithromycin and metronidazole, which has steadily eroded the success rate of standard eradication therapy. Three theories frame the story. The mainstream view holds that H. pylori is an ancient human-associated colonizer whose strain diversity tracks migration. The archaeological reading adds that human crowding, settlement, and changes in sanitation amplified transmission long after prehistory. The more speculative reading suggests that some lineages may once have offered benefits, perhaps immune training or acid regulation, before modern conditions shifted the balance toward disease. A prehistoric stomach passenger became one of humanity's most important cancer-causing microbes. And it raises a question: if a bacterium this old still hides inside roughly half of us, what about a virus that we can sequence directly from a 7,000-year-old skeleton? Number 2. The 7,000-year-old hepatitis B genome from Karsdorf. In a Neolithic burial site in Karsdorf, Germany, archaeologists recovered the remains of a male individual who died roughly 7,000 years ago. He was about 25 to 30 years old at death. What made the find remarkable was not the skeleton itself, but what scientists pulled from his teeth and skeletal material. They recovered viral DNA. Specifically, they recovered hepatitis B virus DNA, in what stands today as one of the oldest sequenced human viral genomes on record. Teeth matter for a reason. Dental pulp is a vascular tissue encased in mineralized enamel and dentin, and it can preserve fragments of blood-borne pathogens long after soft tissues have disappeared. The pulp acts almost like a sealed time capsule. The Karsdorf sequence is part of a larger body of work, including studies led by researchers such as Ben Krause-Kjura and Johannes Krause, that has begun to extract ancient hepatitis B genomes from prehistoric remains across Europe. Hepatitis B itself is a strange virus. It is a partially double-stranded circular DNA virus, but it replicates through reverse transcription. That is, it uses an RNA intermediate, the same general strategy used by retroviruses despite being classified as a DNA virus. The ancient genomes complicate any simple evolutionary story. Some old lineages appear extinct or replaced. Prehistoric HBV was not merely a primitive version of modern genotypes. There's been turnover, with entire branches of the viral family tree dying out while others diversified. Modern transmission happens through blood, sexual contact, and perinatal exposure. Mother-to-child transmission during birth is a particularly important route because it leads to lifelong chronic infection in a high percentage of cases. Chronic hepatitis B can remain silent for years while it slowly damages the liver. The progression moves from chronic inflammation to fibrosis, then cirrhosis, and in some patients to hepatocellular carcinoma. Older estimates put global hepatitis B mortality at around 820,000 deaths per year. More recent figures from 2022 raise that to roughly 1.1 million deaths annually, reflecting either a worsening burden or improved measurement, depending on how the data is read. The combined chronic hepatitis B and C burden exceeds 300 million people worldwide, and a large portion of those infected do not know they carry the virus. The mainstream theory holds that hepatitis B has circulated in humans for thousands of years, with both extinct and surviving lineages. The archaeological reading emphasizes that dense Neolithic communities with their blood contact during butchery, frequent childbirth, and tight kin networks may have sustained transmission. The speculative theory proposes that the prehistoric viral diversity we observe today represents repeated animal to human host shifts that are only partly visible in the genetic record. The Karsdorf genome shows that an infection sitting silently in a Stone Age liver still drives cirrhosis and liver cancer in the 21st century. But not every prehistoric pathogen needs a human host to survive across millennia. Some simply wait in the dirt. Number 3. Tetanus, Clostridium, Tetanus, eternal soil spores. Imagine an ancient wound, a puncture from a sharpened bone, a thorn, or a fragment of stone. Soil contamination, no antibiotics, no vaccine. Days later, the first muscle spasms begin. Tetanus does not need civilization to exist. It needs only a wound, a bacterium, and the absence of oxygen. Clostridium tetani is an anaerobic, spore-forming bacterium. Its spores live in soil, dust, manure, and contaminated environments around the world. They follow human activity wherever wounds, animals, dirt, and childbirth meet. Ancient medical writings describe symptoms that closely resemble tetanus. Texts in the tradition of the Edwin Smith Papyrus, dated to around the 2nd millennium before the Common Era, contain references to lockjaw and rigid spasms following injury. The disease is, in practical terms, prehistoric. Spores germinate only when oxygen levels drop, which is why deep punctures and necrotic wounds are particularly dangerous. The spore itself is a survival capsule. It can persist for years or decades in harsh environments, dormant but viable, waiting for the right anaerobic pocket inside a host. Tetanus is not transmitted between people. The infection always begins with a wound, never with a cough or a handshake. The killing mechanism is a single neurotoxin called tetanospasmin. Once the bacterium grows in the anaerobic environment of damaged tissue, it produces this toxin, which then travels through peripheral nerves toward the central nervous system. There, it blocks the release of inhibitory neurotransmitters, particularly gamma, aminobutyric acid, and glycine. With those brakes removed, motor neurons fire uncontrollably. The clinical picture follows. Trismus, the jaw locking shut, rigidity throughout the body, the involuntary grimace called risus sardonicus, and the backward arching posture known as opisthotonus. Autonomic instability follows. Blood pressure spikes, heart rhythms become erratic, sweating intensifies, and respiratory failure threatens. Surviving the disease does not produce reliable natural immunity, which is one of the strangest features of tetanus. Recovery from infection does not protect against future episodes. Modern prevention depends on the tetanus toxoid vaccine, which uses a chemically inactivated version of the toxin to train the immune system. Boosters are required because vaccine-induced protection wanes over time. Neonatal tetanus, transmitted through contaminated umbilical wounds during unsafe deliveries, remains a major concern in regions without consistent maternal immunization. Roughly 25,000 neonatal tetanus deaths occurred in 2018, a dramatic reduction from prior decades, but still a substantial toll. The mainstream theory states that tetanus persists because environmental spores cannot be eradicated. The archaeological reading argues that agricultural life, animal manure, metal tools, warfare, and childbirth combine to increase exposure across human history. There's no compelling fringe theory here, because the biology is too unambiguous. The soil itself is the reservoir, and if the soil is one ancient enemy, water is another. Number 4. Vibrio cholerae, the pre-agricultural killer born in the Ganges Delta. Picture a river delta, warm brackish water, plankton blooms, monsoon flooding, human settlement along the banks, and invisible bacteria moving through the ecosystem. Vibrio cholerae is an aquatic, gram-negative, comma-shaped bacterium that lives naturally in coastal and estuarine waters. The evidence for prehistoric cholera is more cautious than for some of the pathogens already covered. Direct prehistoric cholera DNA is limited. What is well established is that the organism has an ancient aquatic ancestry, and that its modern pandemic lineages are historically and genomically traceable. The pandemic forms are toxigenic Vibrio cholerae of the O1 and O139 serogroups. The Ganges Delta and the Bay of Bengal are the historic hotspot for cholera emergence. 7 recognized cholera pandemics have swept across the world, with the 7th beginning in 1961 and continuing to the present day. The dominant form during this 7th pandemic is the L-Tor biotype. Cholera's deadliness comes from a borrowed weapon. The genes for cholera toxin are carried by a bacteriophage called CTX-5. Horizontal gene transfer, the process by which bacteria acquire DNA from outside their own lineage, transformed an aquatic microbe into a global killer. The toxin itself, once released in the gut, activates an enzyme called adenylate cyclase, which raises intracellular levels of cyclic AMP. That biochemical change drives massive chloride secretion across the intestinal epithelium, and water follows the chloride. The result is rapid, voluminous, watery diarrhea, classically described as rice water diarrhea because of its pale, flecked appearance. Severe cholera can kill within hours through dehydration and circulatory shock. The World Health Organization estimates between 1.3 and 4 million cases per year, with deaths ranging from 21,000 to 143,000 annually. The wide range reflects the difficulty of surveillance in outbreak settings. 3 theories overlap. The mainstream model holds that pandemic cholera is driven by toxigenic lineages emerging from aquatic reservoirs. The archaeological reading argues that agriculture, irrigation, trade routes, religious pilgrimage, and dense cities turned local water microbes into global threats. The speculative theory suggests that ancient Deltic cholera-like outbreaks may have shaped early settlement patterns long before written records, even though they left no direct genomic trace. Cholera is a disease of water management. But not all ancient pathogens hide in soil or rivers. Some hide in clothing. Number 5, Bartonella quintana, trench fever's 4,000-year-old French burial. A tooth from a Bronze Age burial in France preserved microbial DNA in its dental pulp. The site dates to roughly 4,000 years ago. The pathogen recovered was Bartonella quintana, a fastidious, intracellular, gram-negative bacterium that today is best known for causing trench fever. The work associated with the team led by Didier Raoult, and including researchers such as Michel Drincourt, helped establish that dental pulp can serve as a reliable archive for blood-borne pathogens. When a person carries a bacterium in the bloodstream, that organism passes through the vascular tissue inside teeth, and the mineralized shell can protect the genetic material long after the body decomposes. The vector for Bartonella quintana is the human body louse. Transmission happens when louse feces, which contain the bacterium, are rubbed into broken skin, often through scratching. Humans are the main reservoir. The 4,000-year-old find connects directly to the long history of human lice, clothing, crowding, and poor sanitation. Wherever those conditions exist, the organism can persist. The disease became famous during the First World War, where it was called trench fever and disabled hundreds of thousands of soldiers across the European front. Symptoms include relapsing fever, sharp shin pain, headache, malaise, and chronic bacteremia, meaning the organism circulates in the blood for prolonged periods. The disease often does not kill quickly, but it disables, removes people from work, and slowly grinds down their health. Severe outcomes include bacillary angiomatosis, a vascular proliferative condition, pileosis hepatis, a liver disorder marked by blood-filled cavities, and culture-negative endocarditis, an infection of the heart valves that does not grow on standard cultures. Diagnosis is genuinely difficult. Standard blood cultures may fail. Confirmation often requires serology, polymerase chain reaction testing, sequencing, or prolonged specialized cultures. Endocarditis cases sometimes require valve replacement surgery in addition to extended antibiotic courses. The disease is not confined to history. Recent cases in cities including New York have involved unhoused populations and have presented with endocarditis, heart failure, renal failure, embolic stroke, and deaths. The mainstream theory states that Bartonella quintana persists wherever body lice persist. The archaeological reading argues that clothing, crowding, warfare, and shelters created repeated lice-borne disease cycles across human history. The speculative theory holds that ancient lice-borne infections may have been common but invisible, because lice and the soft tissue lesions they cause rarely fossilize. A bacterium older than written history still spreads through the same conditions that shaped Bronze Age life. The next pathogen, however, is older still and does not need lice or wounds. It moves through cats and meat. Number 6, Toxoplasma gondii. The cat-borne mind-altering parasite older than humanity. The cat is the final host. The human brain is an accidental archive. Toxoplasma gondii is an intracellular apicomplexan protozoan parasite that infects most warm-blooded animals on Earth. Its definitive hosts, the only animals in which the parasite completes its sexual reproduction, are members of the cat family. Felids shed environmentally resistant stages called oocysts in their feces. Those oocysts can survive for long periods in soil and water. Humans become infected through several routes: contaminated soil, contaminated water, unwashed produce, undercooked meat containing tissue cysts, and congenital transmission from mother to fetus during pregnancy. Global estimates suggest that roughly 30% of humans carry the parasite, with major regional variation driven by diet, hygiene, and cat exposure. Most immunocompetent infections are mild or asymptomatic. After the initial phase, the parasite forms tissue cysts, particularly in the brain and skeletal muscle. These cysts can persist for life. The serious clinical pictures appear in two settings. The first is congenital toxoplasmosis, where infection during pregnancy can cause chorioretinitis, hydrocephalus, intracranial calcifications, and a lasting neurologic injury in the newborn. The second is reactivation disease, which occurs when the immune system weakens. In patients with advanced HIV, in transplant recipients, and in others on immunosuppressive therapy, dormant cysts can rupture, causing toxoplasmic encephalitis. The parasite has a strange behavioral footprint. Studies in rodents, including foundational work by Joanne Webster and colleagues, have shown that infected rats and mice lose their innate aversion to cat odors. The interpretation is that the parasite manipulates host behavior to favor predation by the only host in which it can sexually reproduce. Whether anything similar happens in humans is contested. Observational studies have associated infection with risk-taking behavior, personality differences, and certain psychiatric conditions, but correlation is not causation, and the question remains genuinely unresolved. Ancient Egyptian mummy metagenomic studies have detected Toxoplasma gondii sequences. A reported case from a roughly 2,000-year-old South African hunter-gatherer skeleton has been described. Although the evidence is less established than for hepatitis B, the mainstream theory frames Toxoplasma as an ancient cat-prey parasite that humans acquire accidentally. The archaeological reading argues that cat domestication, grain storage, rodents, and permanent settlements increased human exposure. The speculative theory holds that subtle behavioral effects may have influenced human risk patterns, though the evidence remains unresolved. A parasite that travels through cat litter, lamb chops, and unwashed lettuce. Hardly seems like a candidate for biblical drama. But the next infection has been linked controversially to one of the most famous outbreaks in religious history. Number 7. Tularemia, possibly the biblical plague of the Philistines. Picture rodents in stored grain. Swelling. Panic. A disease moving through a coastal population. Tularemia is caused by Francisella tularensis, a small gram-negative coccobacillus. Its zoonotic reservoirs include rabbits, hares, rodents, ticks, deer flies, animal carcasses, and contaminated environments. Infection routes are unusually varied. The bacterium can enter through arthropod bites, direct handling of infected animals, inhalation of aerosolized particles, contact with contaminated water, or consumption of contaminated food. Clinical forms reflect the route of entry. Ulceroglandular tularemia, the most common, produces a skin ulcer with regional lymph node swelling. Glandular disease shows lymph node involvement without an obvious skin lesion. Oculoglandular disease follows eye contamination. Oropharyngeal disease results from ingestion. Typhoidal tularemia is a systemic illness without localizing signs, and pneumonic tularemia, the most dangerous public health scenario, follows inhalation. The infectious dose by aerosol is extraordinarily low, sometimes cited at as few as 10 organisms. That low dose, combined with the disease's severity, is why Francisella tularensis is classified as a Tier 1 select agent in the United States and was studied or weaponized in Cold War biological weapons programs. The bacterium persists in carcasses, water, and cold environments, which adds to its concern profile. In 2007, the researcher Ciro Igino Trevisanato published a hypothesis suggesting that the plague described in 1 Samuel chapters 5 and 6, which afflicted the Philistines after their capture of the Ark of the Covenant, may have been tularemia rather than a bubonic plague. The biblical clues include tumors or swellings, the prominence of rodents in the narrative, the epidemic spread, and the possibility of contaminated grain stores. Tularemia and bubonic plague are competing explanations, and the evidence is literary rather than genetic. No ancient Francisella tularensis DNA has been recovered to confirm the Philistine plague theory. Untreated severe disease, especially the pneumonic and typhoidal forms, can be fatal. Treatment involves aminoglycosides such as streptomycin or gentamicin, fluoroquinolones such as ciprofloxacin, or tetracyclines such as doxycycline. The mainstream theory describes tularemia as a zoonotic spillover disease maintained in wildlife and arthropod cycles. The archaeological readings suggest that ancient rodent grain human systems could have produced tularemia-like outbreaks. The speculative theory holds that the Philistine plague may preserve a literary memory of tularemia, although proof remains absent. A possible biblical pathogen is one thing. A pathogen that may have lived inside Neanderthals is another. Number 8. Brucellosis. The Pleistocene, cave bear zoonosis. The dramatic title needs immediate qualification. The strongest evidence for prehistoric brucellosis points not to confirmed cave bear DNA, but to Neanderthal and early livestock contexts. The zoonosis angle holds. The cave bear specifically does not. Brucellosis is caused by several Brucella species, with the most important human pathogens being Brucella melitensis, Brucella abortus, Brucella suis, and Brucella canis. These organisms naturally infect goats, sheep, cattle, pigs, and dogs. Transmission to humans occurs through direct contact with animal tissues, particularly birth products, through inhalation of aerosols, and through consumption of unpasteurized milk and cheese. Person-to-person transmission is rare. The clinical picture is famously vague. Symptoms include undulating fever, drenching sweats, fatigue, malaise, weight loss, and joint pain. Brucellosis is sometimes called a stealth disease because it is chronic, relapsing, and difficult to diagnose. Complications include osteoarticular disease affecting bones and joints, epididymo, orchitis affecting the male reproductive tract, endocarditis affecting heart valves, and neurobrucellosis affecting the central nervous system. Possible vertebral brucellosis has been described in a Neanderthal skeleton, with spinal lesions consistent with the disease. The interpretation is cautious because spinal lesions overlap with tuberculosis, trauma, and degenerative conditions. The Pleistocene exposure pathway is biologically reasonable. Hunters and scavengers handled animal carcasses, extracted bone marrow, butchered organs, and consumed raw or partially cooked tissue. Stronger genetic evidence comes from later periods. Ancient DNA recovered from an approximately 8,000-year-old sheep bone has revealed Brucella melitensis sequences, placing the pathogen firmly within early herding societies. Brucellosis remains one of the world's most common bacterial zoonoses. Older estimates put the number of new human cases at more than 500,000 annually, with substantial undercounting in regions where laboratory diagnosis is limited. The mainstream theory argues that brucellosis became entrenched through livestock domestication and dairy systems. The archaeological reading proposes that Pleistocene hunters likely encountered Brucella-like infections before farming began. The speculative theory holds that repeated ancient spillovers from wild megafauna may have seeded the lineages that later adapted to domestic livestock. Carcass handling is one ancient route to infection. Skin contact and sexual contact are another, and the next disease has been argued about for more than 500 years. Number 9. Pre-Columbian treponemal disease. The 6,000-year-old syphilis bone trail. The bones tell part of the story. Bowed tibiae, skull lesions with characteristic pitting, deformed long bones in young adults. But naming a disease from a skeleton is harder than it looks. Treponemal diseases include syphilis, yaws, Bigel, and pinta. Syphilis, yaws, and Bigel are caused by subspecies of Treponema pallidum. Pinta is linked to Treponema caretium. The tissues affected include skin, bone, cartilage, nerves, blood vessels, and the developing fetus. Two main hypotheses have shaped the debate. The Columbian hypothesis argues that venereal syphilis reached Europe from the Americas after 1492, traveling with returning expeditions. The Pre-Columbian hypothesis argues that treponemal disease already existed in the Old World before that date, perhaps in different forms. The debate persisted for centuries because skeletal lesions overlap between the various treponemal diseases, and dating problems in older burials can distort claims about pre-1492 presence. Ancient DNA has reshaped the conversation. A reported 5,500-year-old Treponema pallidum genome from Colombia, along with related work by researchers including Verena Schoenemann and Kurtu Majander, has provided molecular support for deep pre-Columbian treponemal diversity in the Americas. That finding does not automatically prove modern venereal syphilis began there. It does suggest that the treponemal family was already complex and geographically dispersed long before European contact. Yaws, the non-venereal treponemal disease that affects skin, bone, and cartilage, particularly in tropical regions, complicates the picture further. Many ancient lesions could represent yaws or Bigel rather than venereal syphilis. The modern burden remains substantial. The World Health Organization estimates that approximately 8 million adults aged 15 to 49 acquired syphilis in 2022, with 1.1 million pregnant women infected during pregnancy. Congenital syphilis, transmitted from mother to fetus, can cause miscarriage, stillbirth, neonatal death, and lifelong disability in surviving children. The mainstream theory suggests that modern venereal syphilis may have intensified after 1492 amid global contact, but did not necessarily originate there. The archaeological reading argues that older treponemal diseases existed across multiple regions of the world before modern syphilis took its current form. The speculative theory holds that ancient climate, migration, and social behavior may have shifted one treponemal form into another, with the venereal route emerging or expanding under specific conditions. The bones cannot fully resolve the debate. The genomes help, but only partially. Some ancient pathogens, however, leave a clearer trail because they integrate themselves directly into our cells. Number 10. HTLV1. The retrovirus that maps the human diaspora. A retrovirus hides inside T cells. It does not produce dramatic outbreaks. It moves through generations, through breastfeeding, through kinship networks, through the slow currents of human migration. HTLV1, the human Opbite T lymphotropic virus type 1, was the first human retrovirus clearly linked to cancer. It is exogenous, not endogenous, meaning it is acquired during life rather than inherited as part of the human genome. Once inside a host, it persists as proviral DNA integrated into the genomes of infected T cells. Most carriers remain asymptomatic for life. The World Health Organization estimates that roughly 5 to 10% of infected people develop recognized disease. Two clinical outcomes stand out. The first is adult T cell leukemia lymphoma, an aggressive cancer of mature T cells. The second is HTLV1-associated myelopathy, also called tropical spastic paraparesis, a chronic neurologic disease affecting the spinal cord. Transmission occurs through breastfeeding, sexual contact, blood products, and shared needles. Breastfeeding is a particularly important route in endemic family lineages, which is one reason the virus tracks so closely with kinship networks across generations. Compared with rapidly mutating RNA viruses, HTLV1 has a relatively stable genome. That stability makes it useful for tracing human dispersal. Researchers have used HTLV1 phylogeny in the same way population geneticists use mitochondrial DNA or Y chromosome lineages, treating the virus as a marker of historical contact between populations. Ancient HTLV1 proviral DNA has been recovered from an Andean mummy. The work, associated with researchers including Alain Froment and Renaud Boulanger, showed that the ancient sequences resembled modern Andean and Japanese-related lineages. The interpretation is that HTLV1 reached the Andes before colonial contact, supporting deep pre-Columbian circulation in the Americas. Endemic foci today include southwestern Japan, the Caribbean, parts of South America, sub-Saharan Africa, and Australo-Melanesia. The mainstream theory holds that HTLV1 followed deep human migration and kinship-based transmission. The archaeological reading argues that isolation in indigenous Pacific Andean and Australo-Melanesian populations preserved ancient viral lineages that might otherwise have been overwritten. The speculative theory holds that rare ancient ocean crossings, founder effects, and now lost migration corridors may explain some of the puzzling lineage distributions seen on modern phylogenetic trees. 10 pathogens have been laid out. The patterns connecting them are now worth examining. After 10 pathogens, a structural question deserves direct attention. Did these microbes follow humans, wait in the environment, or erupt when civilization changed? The answer is rarely clean. 3 explanations recur. They are not mutually exclusive. The first is co-migration. H. pylori and HTLV1 fit this pattern. Both organisms ride along with human populations across generations, encoding information about ancestry and movement in their genetic structure. Their phylogenies map onto human migration routes with striking fidelity. The second is environmental amplification. Tetanus, cholera, and tularemia fit this pattern. These pathogens existed in soil, water, and wildlife long before they became major killers. What changed was the human side of the equation. Settlement density, water management, agriculture, and animal handling created the conditions under which environmental microbes became epidemic threats. The third is intimate transmission, transformed by human social behavior. Hepatitis B, brucellosis, and the treponemal diseases fit this pattern. Blood, birth, sex, and animal contact were the routes. Domestication, dairy systems, and changes in sexual networks shifted the pathogens from background presence to sustained burden. Prehistoric does not always mean ancient DNA proof. There is a meaningful distinction between direct molecular evidence and ecological plausibility. Hepatitis B and Bartonella quintana stand on strong ancient DNA grounds. Tularemia and cholerae depend more on hypothesis-driven reasoning. Brucellosis is a cautionary case where the dramatic framing has to be tempered by careful reading of the actual evidence. The mainstream origin model emphasizes host ecology, mutation, selection, and transmission networks. The archaeological model adds settlement, agriculture, warfare, trade, sanitation, and domestication as amplifying forces. The speculative model considers whether ancient myths, biblical plagues, and unexplained skeletons may preserve distorted memories of infections that left no other trace. These theories overlap rather than cancel each other. A pathogen can be ancient while its modern disease pattern is recent. Direct proof remains rare. DNA decays, soft tissue disappears. Many infections leave no bone lesions at all. Teeth, the petrous bone, dental calculus, and mummified tissue have become disproportionately important precisely because they preserve what other tissues cannot. Ancient genomics is rewriting old medical history. The words possible, suggestive, and confirmed must be kept distinct. With that frame in place, the methods themselves are worth understanding. Most infections do not fossilize. That is the central problem. Paleopathology, the practice of reading disease in bones, provides one set of clues. Treponemal diseases leave characteristic skull and long bone lesions. Brucellosis can produce vertebral changes. Some chronic infections leave traces in dental calculus or in healed fractures. But bones are unreliable witnesses. Different diseases can produce similar damage. Tuberculosis, syphilis, brucellosis, and chronic osteomyelitis can be hard to distinguish from skeletal evidence alone. Ancient DNA has become the stronger modern tool. Teeth preserve blood-borne pathogen DNA inside the dental pulp, sealed within mineralized enamel and dentin. The petrous bone, a dense region of the skull near the inner ear, is one of the best sources of well-preserved ancient human DNA, and it can also yield pathogen sequences. Two main approaches dominate. Metagenomics involves sequencing all DNA present in a sample and then computationally identifying microbial fragments. Targeted capture uses synthetic probes to pull out specific pathogen sequences from highly degraded material, dramatically improving sensitivity for known organisms. Authentication is critical. Ancient DNA shows characteristic damage patterns, particularly cytosine to thymine substitutions at fragment ends, and tends to come in short fragments. Researchers including Svante Pääbo, Johannes Krause, and Hendrik Poinar have developed protocols for distinguishing genuine ancient sequences from modern contamination. Independent replication in separate laboratories is part of the standard. Phylogenetic analysis then places ancient pathogens onto trees alongside modern lineages. H. pylori and HTLV 1 provide examples of phylogeographic inference where geographic distribution and evolutionary relationships are read together. Hepatitis B illustrates a different lesson, where ancient lineages do not neatly match modern diversity, indicating extinct branches and replacement events. Bartonella quintana shows what dental pulp recovery can do when the right tissue is available. RNA viruses are harder to find than DNA viruses because RNA degrades more quickly. That fact biases the prehistoric viral record toward DNA pathogens. Sample bias matters. Mummies, cold graves, dry caves, and elite burials are overrepresented in the literature simply because they preserve material better. Absence of evidence is not evidence of absence. Modern reference databases can mislead if ancient contamination is not controlled, since modern microbial DNA is everywhere in the laboratory environment. Every ancient infection claim deserves a confidence level. With the methods understood, the question shifts from how scientists find these pathogens to why they still kill. The paradox is straightforward. Old infections survive inside modern systems with vaccines, antibiotics, sanitation, and intensive medical care. Each pathogen exploits a specific kind of reservoir. Tetanus exploits environmental reservoirs. Spores in soil. Cholera exploits aquatic reservoirs. Vibrio cholerae lives in water systems whether or not humans are sick. Brucellosis exploits animal reservoirs. Cattle, sheep, goats, and pigs maintain the bacteria regardless of human medical infrastructure. Bartonella quintana exploits a vector reservoir. Body lice persist wherever conditions permit them. Helicobacter pylori exploits a household reservoir. Childhood transmission within families keeps the bacterium moving across generations. Hepatitis B and HTLV-1 exploit lifelong reservoirs. Chronic carriage in the human body itself is the storage system. Toxoplasma exploits a latent tissue reservoirs. Cysts in muscle and brain persist for decades. Congenital transmission carries some of the most ancient pathogens directly into the next generation. Hepatitis B, syphilis, and toxoplasmosis can all pass from mother to fetus. Silent infection is its own survival strategy. Asymptomatic carriers keep pathogens moving long before disease is recognized. Poverty operates as a biological amplifier. Crowding, sanitation gaps, unsafe water, and limited access to care all increase transmission. Conflict and displacement accelerate cholera and lice-borne disease. Low vaccination coverage opens the door for tetanus and hepatitis B. Antibiotic resistance, particularly in H. pylori treatment, blunts the tools that worked a generation ago. Diagnostic weakness affects brucellosis, Bartonella, and HTLV-1, where the available tests are limited or require specialized capacity. Eradication is impossible for some pathogens because their reservoirs lie outside humans. A disease confined to a single human host species, like smallpox, can in principle be eliminated. A disease maintained in soil, water, livestock, or wildlife cannot. Modernity removes some risks while creating others. Industrial food systems can amplify Brucella and Toxoplasma. Mass travel can spread cholera. Urban inequality can recreate the conditions that supported Bartonella in the trenches. The microbes survive by occupying the cracks in civilization. Each crack corresponds to a specific failure of public infrastructure. Each ancient infection points to a weak spot in human infrastructure. The list is essentially a public health checklist written backward. Helicobacter pylori reveals the limits of antibiotic eradication and the cancer prevention opportunity attached to it. Hepatitis B reveals the importance of birth dose vaccination, since perinatal transmission produces the highest rates of chronic infection. Tetanus reveals the importance of maternal vaccination and sterile delivery practices, particularly in low-resource settings. Cholera reveals the centrality of clean water, sanitation, and rapid rehydration capacity, since oral rehydration therapy can save lives within hours when it is available. Bartonella quintana reveals the importance of housing, laundry access, and louse control. Toxoplasma reveals food safety practices, prenatal screening, and basic hygiene around cat feces. Tularemia reveals wildlife exposure patterns, aerosol risk, and the need for biodefense surveillance in the modern era. Brucellosis reveals the value of livestock vaccination, milk pasteurization, and slaughterhouse safety. Treponemal disease reveals sexually transmitted infection testing, prenatal care, and access to penicillin. HTLV1 reveals breastfeeding counseling in endemic regions, blood product screening, and surveillance of so-called neglected viruses. Some controls are simple but unevenly distributed. Oral rehydration solutions are inexpensive, but they have to reach patients within hours. Tetanus toxoid is one of the cheapest vaccines ever made, but coverage requires consistent immunization programs and booster maintenance. The hepatitis B vaccine works extraordinarily well, but missed birth doses allow lifelong infection in the children who should have been protected from day one. Penicillin treats syphilis effectively, but missed prenatal screening leads to congenital cases that should never occur. Brucellosis control works best when the focus is on animals before humans become infected. Bartonella prevention requires social infrastructure, not just prescriptions, because lice will return wherever housing collapses. Public health work in this domain operates almost like archaeology in reverse, removing the conditions that allowed ancient pathogens to thrive in the first place. Prevention is the modern version of breaking prehistoric transmission chains. The closing question is what general principles emerge from the whole list. The body has been mapped repeatedly across this script. Stomach, liver, nerves, blood, brain, bone, immune cells. Each pathogen exploits a particular feature of human biology and a particular feature of natural law. H. pylori is chemistry mastery, surviving acid through urease-driven ammonia production. Hepatitis B is genetic persistence, combining DNA storage, reverse transcription, and lifelong carriage to outlast immune responses. Tetanus is environmental durability, with spores waiting in soil for decades until an anaerobic wound provides an opportunity. Cholera is water physics, where fluid loss converts a toxin into mass mortality through simple osmotic mathematics. Bartonella is vector dependence, with lice thriving wherever hygiene collapses. Toxoplasma is predator-prey ecology redirected into accidental human exposure. Tularemia is low-dose zoonotic danger, where 10 organisms can be enough. Brucellosis is the cost of animal domestication and food systems. Treponemal disease shows the difficulty of separating sex, skin, bone, and history when reading the past. HTLV1 is migration recorded directly in viral inheritance. None of these pathogens needed modernity to exist. They were already here in soil, water, animals, and human bodies. What modernity added was infrastructure that the pathogens could exploit. Cities, trade, hospitals, war, displacement, industrial food systems. Each new system created new opportunities for transmission. Science counters them with the same natural laws that the microbes use. Vaccination is controlled immune memory. Antibiotics are chemical interference with microbial survival. Clean water is ecological interruption. Pasteurization is heat applied to microbial vulnerability. Genomics is reading evolution directly and using that reading to guide intervention. Innovation is not escaping nature. It is learning the rules well enough to stop ancient infections from using them against us. The prehistoric pathogens covered here did not die out when civilization arrived. They adapted to it, and in some cases, they grew stronger. The work of public health, paleopathology, ancient genomics, and clinical medicine is to keep adapting in turn. Every spore, every cyst, every chronic carrier represents both an evolutionary success story and a problem still waiting for the right combination of vaccine, antibiotic, sanitation, housing, and surveillance. The deadliest prehistoric infections are not relics. They are ongoing experiments, and humans are still part of the data set.